Unit 2, Lesson 3, Guided Notes, Meiosis. Now, just like we said in the introduction, this is going to be a little bit more complicated and a lot more drawing. So make sure that we copy down the descriptions first, and then we'll tackle the drawings. So let's talk about meiosis. Meiosis is a form of sexual reproduction, and this is a form of cell division that's specific to form sex cells, or what we also call gametes. At the end of it, it's a half cell that's being made, and we use the term haploid. And so in this process, we take a diploid cell that goes to a haploid cell, and that there's two divisions, meiosis 1 and meiosis 2. So take this again, note of this. You don't have to write the two divisions, meiosis 1 and meiosis 2, because those are actually already in your notes for you. Fill in the blanks with diploid and haploid, but do write the description that's in orange for your description of sex reproduction equals meiosis. Let's go and get started. So interphase, this is right above your chart. We're going to write really, really small. It's similar to mitosis in interphase. That chromosomes are replicated in S phase, just like interphase in mitosis, and that each duplicated chromosome consists of two identical chromatids attached at their centromeres. And again, what I have under bold is the really important parts. Now, once we enter our table, and remember, you can pause this whenever you need more time to write. We're starting meiosis 1. So meiosis 1, specifically prophase 1. If it has a number, that means it's a part of meiosis. Now, in prophase 1, chromosomes condense. Homologous chromosomes form a tetrad. Now, a tetrad is two chromosomes of four chromatids. And crossing over occurs here. We're not going to define crossing over here. It's defined later on, but it's really the exchanging of fragments, and it's the first part of genetic variation. It's the first part of what makes these cells unique instead of identical. Now, as you can see in these different pictures, you have the chromosomes represented by the X's, that they get really close together. Synapsis occurs. You don't need to know this. You don't need to write this. And that they exchange fragments. So you can see where the colors, we use colors a lot of times to represent this happening, switch over. Now, if we're going to try to draw this, I've included the drawing that I include in my notes. First thing, I just write this extra part of 2n equals 4. We're starting as diploid. I just draw, this is in my box. I draw a circle. Within my box, I draw a dotted line. We represent this dotted line just like we did in mitosis as a nucleus is disappearing. I draw my T's. You don't have to draw them like this. You can just draw a T. A T with lines in between to show that the spindle fibers are forming. And then I draw my X's, and I like to draw a total of 6, but you can see how they're paired up and they're of similar size. Now, you can come back later and do the coloring. You don't have to do that now. That's going to take a lot of time, and that can maybe make your notes a little bit better. Might be a good way to sort of just study for this at a different time. So you have these pairs of chromosomes, and they cross over and exchange fragments. We have crossing over that we write in there. Now, with meiosis 1, metaphase 1, so we're now going to P, mat, so M, metaphase. Tetrads line in the middle. Remember, metaphase, middle. And we use the word tetrads because they're still sort of paired up from where they sort of exchanged it. And here's the next part of genetic variation. It's the second and only other part that we have that's specific to genetic variation. Independent assortment occurs here, and it's random. What that means is, let's say this, look at this picture over here. See how we have the pink and up on the top and the purple on the bottom? It's completely random that that's how it forms. The purple could have very easily been on the top instead. Again, it's random, and that's how it's sorted. And this is part of why, for instance, with siblings, you guys don't look exactly alike. Same thing with what happens with crossing over too, of course. Now let's draw it. Again, we have our circle. You can actually probably add another spindle fiber right here to go to these two homologous chromosomes. I draw my centromeres, I draw my spindle fibers, I draw my X's lined up in the middle. I like to draw a dotted line to definitely know, hey, I'm lined up in the middle. I put the random assortment is occurring. And again, you can color this later, but just make sure that you have your X's. Now, anaphase 2. This is where we have homologous chromosomes separate. So they separate and they're pulled apart. So remember, anaphase apart or anaphase away. So homologous chromosomes separate, but sister chromatids stay together. So these chromosomes are these chromatids aren't pulled apart yet. They're still attached at their centromeres. All right, when we're drawing this, again, we have them pulled to the poles. They're pulled apart. We draw our cell, we draw our T, we draw our spindle fibers attaching, and then the X is getting pulled apart. Again, you can add your colors later. So meiosis 1, telophase 1. This is the last phase of meiosis 1. Now with this one, each side now has a haploid set of non-identical chromosomes. Cytokinesis occurs. That's the cutting of the cell. And the non-identical haploid parent cells are formed. We're not done yet, but we're getting there. Now, as you can see, we start to see that nucleus, the nuclei forming again. And then the process is going to start all over because now we need to work with two separate cells. Now let's draw this. So as you can see, I have my splitting of my cells. You can see where I have the dotted lines that I drew for my nuclei reforming. My spindle fibers are going back in, and I have my chromosomes, sister chromatids in each individual nuclei that's forming. Now, at the very bottom of this page, you have a fill in the blank. Cytokinesis occurs here. The cell splits into two haploid, non-identical, unique cells. So write that into your notes.